It was astounding to me. It absolutely woke me up and opened my eyes. I saw for the first time that disease susceptibility is not just a problem of the genome, it's a problem also of the software, which we call the epigenome.
Genetics explains about 5 to 10% of most of these diseases we see, but epigenetics has the potential to explain a whole lot more.
We were kind of outcasts, kind of weird autistic exceptions who were working on something that nobody thought was important.
Because of the aging population, neurodegeneration is becoming a huge issue. We're right at the beginning of understanding the role of the epigenome in various types of brain disease. So you could imagine with epigenetic drugs, we might be able to delay or prevent the development of Alzheimer's.
The thought is that if we understand this epigenetic code, maybe we can reset our body to a point before we started getting the onset of diseases or the problems that kind of accumulate with age.
We don't know what impact our discoveries will have. We really don't know. Even the guys who make the discoveries don't appreciate the impact.
At the start of the millennium, scientists cracked the code of the human genome. This discovery took thousands of scientists more than 10 years and $3 billion to complete. It was believed that this was the genetic blueprint to the Book of Life.
So we had the Human Genome Project basically wrapped up about 2003. We thought that if we understood the genetic code, we'd have the answer for all the causes of all known diseases. But it turns out it's a lot more complicated than that. And here along comes the people interested in epigenetics and basically burst the doors open to the dam by suggesting that the environment can directly impinge upon the epigenome. Then it became a lot more complicated.
One of the people responsible for breaking the dam wide open was cancer biologist Randy Jirtle. When he stumbled upon a tumor suppressor gene that was controlled by environmental factors and not genetics, Jirtle realized that scientists had been missing half the picture.
It totally took me out of the genome world, which is where still most of the scientists are, and forced me to think about the problem of disease susceptibility from an environmental standpoint. So it was clear to me that one of the most important areas in biology at that point was going to be the study of epigenetic regulation of gene expression.
Epigenetics plays a crucial role in regulating biological processes by chemically modifying our DNA without changing our genes. Every single cell in the human body has largely identical DNA. The DNA in each of these cells is 2 meters long. To fit into the tiny space inside each cell, DNA is packed down into a microscopic size using packaging material. Through a process called DNA methylation, little chemical tags called methyl molecules attach to this packaging material to turn the genes on and off. If the gene is turned on, it's active and can be read. If it's turned off, it cannot be utilized.
I like to think of the definition of epigenetics in a very simple way, in that I think of the genome as being comparable to the hardware of a computer. And the epigenome, which is above the genome, above the computer, is the software that tells that computer when, where, and how to work. So this is why you have the exact same genome in every cell in your body, but yet you have 260 different cell types in your body doing completely different jobs.
All of the researchers in this kind of epigenetics field, to some extent, we're all kind of like computer programmers. We're trying to understand this code so that we can reprogram and reset the software.
Imagine reprogramming our bodies, rewinding our genetic clocks to eradicate diseases such as diabetes, Alzheimer's, and even cancer.
As we age, we get a lot of insults from the environment, from our diet, stresses that we've had over our lifetime, and also things we've inherited, like mutations that make us susceptible to certain diseases. However, the thought is that if we understand this epigenetic code, maybe we can reset our body to a point before we started getting the onset of diseases or the slowing down or the problems that kind of accumulate with age.
External factors such as the air we breathe, the food we eat, and the stresses we face each day regulate which genes are turned on and off. In 2003, Randy Girdle published a small report that would secure his place in history as the granddaddy of epigenetics. The Agouti mouse study became one of the most cited scientific studies in history.
The Agouti mouse is a perfect model for studying the effects of environmental exposures while these animals are in the womb.
These Agouti mice pups, born to two different gold-coated mothers, are genetically identical. Their differences in size and color were determined by their mother's diet.
The Agouti gene is regulated epigenetically, and you see a coat color readout. Yellow animals become obese, get diabetes and cancer at high incidence, so it's also absolutely linked to diseases, states that are bad. And the brown animals don't have any of these problems, and they're lean and clean all of their life. And this one here, the mother got food that was treated with choline, betaine, vitamin B12.
Simply by changing the mother's diet during pregnancy, Girdle was able to manipulate her offspring's fur color and health without altering a single letter in their genetic code.
And fast, much faster actually than the other one. We had normal mouse diet, one group, and the other group was a synthetic diet that was supplemented in folic acid, choline, vitamin B12, and betaine. The mother would eat them, and as a consequence, the offspring would be more likely to be brown and disease-free. And that indeed is what we showed.
Girdle's study would forever change the way we view disease and ushered in the environmental epigenetics era.
Our research was covered in basically every major newspaper, in a way all over the world.
As a result of the Agouti mouse study, a decades-long mystery would finally be laid to rest. The Agouti mouse study showed that a mother's diet could affect her offspring's genetic code. This shed new light on a mystery that had plagued researchers who had found similar results in children born during the Dutch famine of 1945.
At the end of World War II, the Nazis had still control of part of Holland, and it was a very severe winter, and 20,000 people died during that winter because of starvation. Some of the people that were in that area of Holland during that severe winter were pregnant. So what the mother was exposed to, which was a lack of food, a severe lack of food, and the stress that went with that, ultimately gave rise to offspring that had an increased incidence of cardiovascular disease, obesity, and then other people also showed that these offspring, that particularly if they were in utero during that hunger period, during the first trimester, also had a doubling or so of the incidence of schizophrenia.
Girdle's findings confirmed what the Dutch famine study had suggested.
The placenta perceived that there was this famine, there was a drop in the availability of food, and programmed the metabolism of the fetus that when you're born, when you get out there, if you see food, you better eat it right away, and your body better hang on to it, because there's not much food out there. So this is why they then go on in a culture, in a society of plenty, in post-World War II Netherlands, that they went on to develop diabetes and obesity and so on. So there was a mismatch between the environment that the mother was experiencing during the pregnancy and the external environment that these children and eventually adults were brought up in.
Shockingly, the adverse health effects could even be seen in the grandchildren of those exposed to the famine.
It looks like there's the potential for what's called an epigenetic legacy that can be passed forward from generation to generation. It's really incredible because now you can not only inherit genetic mutations from generation to generation, but you can inherit epigenetic changes from generation to generation. And those changes can either be advantageous or not advantageous, depending on what regions of the genome are being affected.
The idea that what your mother ate or didn't eat could change your heredity without changing your genes was scientific heresy. And epigenetics was met with a great deal of skepticism.
People hated epigenetics. It wasn't like, okay, this is another discipline that I'm not interested in. It always created antagonism.
Montreal geneticist Moshe Saif helped push epigenetics into the forefront by showing that your genes could be also modified by your environment.
I think two discoveries made the big change. One was Randy Gertle's yellow Agouti mice, and the other was our discovery of the maternal care and epigenetics.
Saif's groundbreaking maternal care study would push the envelope further than ever before.
Perhaps the way by which maternal care works is by changing the way DNA is marked. Of course, that was a crazy idea at the time.
Perhaps not so crazy after all. The maternal care study showed that the nurturing behavior of mother rats shaped the epigenome for pups and actually molded their personality traits. The maternal licking stimulated a chemical change in the brain that regulates how much stress hormone is released by the rat's adrenal glands. The less grooming a rat pup received, the more stress hormones it produced in adulthood.
The female pups, whose mother treated them like with low care, they will treat their pups with low care. So what we found was, two things were important. First, that it wasn't the biological mother that was important. It was the caring mother. So you could cross-foster. So the fostering mother did as well as the real mother. So it suggested it's not the genes that go through the germline. So that was the first proof that it's epigenetic. But the second thing is, what's the mechanism? And we found changes in epigenetics, both in what we call chromatin and the histones and the DNA, in the brains of the pups that were treated with high maternal care, that were different than those pups that were treated with low maternal care. And this was the first evidence that the mother leaves a signature, chemical signature in your DNA. And then the next important thing was, it's not final. So even though it's very stable, can go for generations, you can change it. So we can reverse it by using epigenetic treatment.
The idea that epigenetic modifications can occur after birth through behavior and be passed on was a game changer. Over the past 10 years, Saif and his team have applied this research to humans and found that those who'd experienced childhood abuse had different epigenetic markings on the DNA in the brain's hippocampus than those who had not. As epigenetic discoveries continue to provide evidence that we have some control over our genetic legacy, interest in the field has blossomed.
Now people who never thought about, DNA or things like that, like psychologists, psychiatrists, nutritionists, social scientists, political scientists, economists, started getting interested in DNA, because now DNA has life. It's not just a stale thing that you're born with and die with. It's full of life.
Because of its far-reaching effects, epigenetics has garnered the attention of scientists from across many disciplines. This has led to some unusual collaborations. Epidemiologist Catherine Hoyo was so inspired by Randy Gertle's work that she suggested they team up.
When I saw Randy's Agouti mouse study, I thought, this is what I've been looking for. The problem at that time was that there were no studies in humans outside the tissue-specific cancer-related type studies. So the question is then, how do you take something that you see in a mouse and translate it to humans?
Hoyo developed the Newborn Epigenetic Study, or NEST, to look for epigenetic markers for early obesity. She and her team recruited 2,500 pregnant women from the Durham, North Carolina area. They collected blood samples from the women during the earliest stages of pregnancy to determine if, like in the Agouti mice, low folate levels led to obesity in the children.
anything you were exposed to or anything you ate would influence the way that the genes in your child are transcribed, and whether that has anything to do with the outcomes we are looking at, like obesity, like ADHD.
Her research uncovered something quite unexpected. The women had high levels of a neurotoxic metal called cadmium in their blood.
When we found these elevated levels of cadmium exposure, and then I thought, this doesn't look right. So I drove across town to the other side of town where those women live and realized that that area is kind of barren. So then we decided to try and see, do we have cadmium exposure here that seems like it's clustered? And as it turns out, yes, there was, but there was a lot in the soil.
Hoyo suspected that the high levels of cadmium in the mother's blood could be leading to the obesity she was seeing in their very young children.
So then I thought, this is a poorer part of town. There is a fast food restaurant every corner. Is it because of that? Is it the poverty?
After testing her cadmium theory on zebrafish, her suspicions were confirmed.
If you're having an obesity phenotype at age 5, chances are it's going to turn into diabetes or something else later in life. So the studies that we are doing now is to try and look for this epigenetic signature, right? The methylation, the CpG methylation signature that could tell somebody at birth that your child was exposed to cadmium. Because cadmium is not just North Carolina that has this problem.
Studies like Hoyo's provide insight in how exposure to environmental toxins, especially pollution, affect the chemical tags that attach to our DNA and have negative repercussions on our health. The goal is to identify epigenetic signatures at birth and intervene to prevent disease. Once considered outcasts, epigeneticists are now widely respected for their insight into one of the most exciting new fields of science. The Perelman School of Medicine at the University of Pennsylvania is one of the world's top research-oriented medical schools.
So epigenetics is really, it's blossoming around the world, and we've really taken advantage of that here at Penn by creating an institute, Penn Epigenetics Institute. We really run the gamut between basic research all the way to trying to understand all the clinical implications of epigenetics.
We have the largest epigenetics institute in the world. There's 60 to 70 faculty here that all have their own distinct labs studying chromatin biology and epigenetics. This is the epicenter of epigenetics.
Here at Penn State, some of the world's top experts are investigating the role epigenetics plays in age-related disease. And the key to the fountain of youth might just lie in this ant colony.
The queen can live upwards of 20 years, while the workers never live above about a year at the most. The interesting thing about these ants is that any single one of these workers, if you were to isolate it or remove its queen, can become a replacement queen, and they will live up to 4 years. So effectively, they can drastically change their lifespan. Somehow, they're turning on certain genes or mechanisms to make them live considerably longer, and everybody wants to live longer, so.
Epigeneticists believe that solving the mystery of longevity in ants will provide clues to treating age-related disease such as neurodegeneration. Unlike most other cells in our body, neurons, our major brain cells, do not divide.
You can imagine that to create memory, rather than major changes in new cells being generated, you're going to have to change the cells that are already part of the brain. And so it's now believed that epigenetic regulation has a very large part in creating new synapses, creating new circuits between cells to create new memory. What we've discovered is there's a dramatic difference in the way the epigenome looks between older normal brains and Alzheimer's brains. From an epigenetic point of view, we can see that normal human brains have epigenetic protection, if you will.
Berger's studies have shown that Alzheimer's disease is not simply an advanced stage of normal aging, but rather dysregulated aging that changes the structure of DNA.
Now we can develop, for example, an epigenome test to sample the epigenome. If you know there's familial propensity to develop Alzheimer's, you could imagine with epigenetic drugs, we might be able to delay or prevent the development of Alzheimer's.
Epigeneticists are looking once again to ants to find the clues to other brain disorders.
In all ant colonies, you have one or a few individuals do all the reproduction, lay all the babies, so to speak. The brood, that's a queen. Typically people know them as the queen ants, but in this species, the worker ants that do all the work have two different phenotypes. So one is the smaller worker that tends to do most of the foraging and the work here around the nest. And then the other is this real large one that's just trying its best to destroy me, and that's a soldier worker, and these are mostly defense related.
Ants provide ideal models to study social behavior because each colony is comprised of thousands of individual sisters. The queen and all the workers are female, with nearly identical genetic makeup, much like human twins.
What we've been interested in is how does epigenetics work to regulate the forager behavior compared to the soldier behavior? And so what we've been able to do is use some of these epigenetic drugs and literally inject them into the brain of the soldier caste and convert the soldier behavior into forager behavior. She still looks the same. She's big and strong and muscular. So now she goes out and forages for food, which takes more stimulation of the brain to be able to do that.
The idea of using epigenetics to reprogram behavior could have huge implications in humans. Researchers at the University of Pennsylvania have been able to completely reprogram the behavior of ants using epigenetics. These findings could have far-reaching implications for treating our soaring mental health crisis.
One area that my lab is very interested in is addiction memory and PTSD. These are two areas where there are unwanted memories. With the opioid epidemic, the idea that we may be able to lower trauma memory is a really exciting area that my lab's become very interested in.
Epigenetics explains actually why addiction is so hard to treat. We got an addictive app, and we have to get rid of that app. And the way you get rid of an app in a computer is you reprogram or you erase it. And I think we can do the same with epigenetics. I believe that if we reprogram, it will never come back. On the other hand, if we just treat with the symptoms of addiction, we'll never cure it, because the underlying cause is deep in our DNA, and this is what we need to take care of. We have to solve this problem. An addicted person is a ticking bomb. We know that. And I think using epigenetic strategies, even epigenetic rebooting, to kind of get rid of that program is worthwhile.
Our environment affects the way our genes are expressed and can have long-term implications for our health. One of the most pervasive environmental factors proven to have a detrimental impact on our genes is stress.
Not only can environmental factors like nutrition and exposure to toxicants and physical agents like radiation and stuff potentially alter the epigenome and, as a consequence, change your disease susceptibility, but stress is also a factor that can do the same thing. And in humans, where you see this is when you have natural disasters.
The long-term effects of stress are still evident 2 decades after one of Canada's largest natural disasters, the Quebec ice storm of '98.
The ice buildup on the high tension lines, the weight of the ice on these power lines was so great that it toppled even these huge structures carrying electricity. And so there were 3 million people without electricity. The temperatures then dropped to -10, -20. So people were without electricity for a very long time and unable to heat their homes, having to scramble to keep their family safe and warm.
It was like a war zone. All the electric pole were down, electric wire was down. When you say no electricity, it means no gas station, no bank, no grocery store, nothing of that. So if you want something to eat or something like that, you don't have anything around here. So that was pretty scary.
Suzanne King is a social psychologist at the Douglas Mental Health University Institute in Montreal. She's been leading a study on the effects stress from the ice storm had on pregnant women and their children.
In the Project Ice Storm, those women were without power for an average of 14 days, and some for as long as 45 days. So we were able to really tease apart objectively what happened to these moms. What was their loss like, their level of personal threat, the scope, so how long did it last, and how much did their life change? When we started actually assessing the children directly, we had about 150 families in the study. At this point, 20 years on, we have about 60 children still left in the study.
One of the 150 pregnant mothers taking part in the study is Julie Groslo. Groslo was in her 3rd trimester at the time of the ice storm. Her son Vincent is now 20.
It was pretty long, 20 long day. You want the last day of your pregnancy to be beautiful, or you want to be ready, but no, it wasn't like that too much. Even at the hospital, when I gave birth to Vincent, in the night, they were shut down because the generator have to pass the generator by generator, so they have some shutdowns. So you're in the hospital and poof, it's long when you don't know what's happening. It was pretty scary.
The children who took part in the study were followed and tested over many years. An analysis of blood and saliva samples taken from the children at age 13 astonished researchers. More than a decade after the ice storm, the children had high levels of DNA methylation in their blood.
So we're showing how the DNA methylation, how this epigenetic change is then translating into something that we can observe in the children. I've been very struck by the objective severity of the hardship imposed by the ice storm having massive effects on cognitive development and the physical development. IQ, memory, language development, also their body mass index, their immune function, their insulin secretion. We can see that the more days without electricity, for example, the lower the IQ in the children. In terms of the mom's subjective distress from the ice storm, this predicts the children's behavioral development, so anxiety, depression, also their autistic-like symptoms at age 6.5, but not much else.
Here is our first meal at the restaurant. In here, that's Vincent. He had a lot of allergy, food allergy, allergic to milk, to peanuts and seafood, and there's no allergy in my family. Only Vincent have allergy. He have some gastric reflux too. He have laryngomalacia. He's doing a lot of noise when he's breathing. And when he was young, at school, he worked very hard. He always worked hard. He always worked hard for his speaking. He have some speaking problem, and my oldest son doesn't have anything of that. So it's not my gene, it's not the gene of my husband or my parents. Why he have all those things? This is Vincent. This is girlfriend, Valérie, who was also born in the ice storm. I'm proud of him because now, like a carpenter, he's very good. He loves his work. So even if he has some problem, young, now he's living a life, a normal life.
The Quebec ice storm was one of Canada's most devastating natural disasters, but it pales in gravity to other larger disasters.
If you think about some of these much more life-threatening disasters like earthquakes or tsunami, those kinds of disasters, I'm just imagining that the effects must be that much greater.
Epigenetics is shedding new light on one of our most complex public health challenges, the obesity epidemic. More than 2 billion people now suffer from serious health problems related to being overweight.
Obesity rates have skyrocketed. Health consequences as a function of that are estimated to be, in the billions of dollars worldwide, including in not just in the West, but in developing countries as well. So it's a worldwide phenomenon.
A deeper insight into the mechanisms at play in obesity may be hidden in our epigenome. When a mother experiences stress during pregnancy, her unborn child is epigenetically programmed to read that stress as lack of food.
That's how it used to be for thousands and thousands of years of human evolution. But now this is not necessarily so, right? So poor people have access to food, especially in the West. And so those genes that will protect you from starvation, because they will make you binge, can now become harmful. So what you want is to identify those people who will be at risk of a mismatch, who have an epigenome that prepares them for a life of nutritional deprivation, but we know they're going to live in the United States or Canada or Western Europe, where food is not expensive. And therefore, could we devise intervention strategies? They don't have to be drugs. They could be behavioral strategies that will teach people who are naturally prone to binge that binging is not necessary anymore because we're living in a very different world.
Biotech and pharmaceutical companies worldwide are making steady progress in the development of epigenetic drugs to treat some of mankind's most dreaded disease. Epigenetics has recently emerged as the most exciting and hopeful new weapon in the war against cancer. Mojay Saif has been there from the beginning, challenging the entrenched beliefs.
I remember meeting with a very prominent cancer scientist when I was a graduate student, and I asked, do you think that, epigenetics has something to do with cancer? And he said, it's all genetics. There's nothing epigenetic in cancer. And these were the days when, the first cancer genes were discovered. Everybody was very excited about it. The guys who discovered it were all pop stars, and to say that something else might be involved was like kind of sacrilegious. And then we went deep into the biochemistry of DNA methylation, and we saw that it is being rearranged in cancer.
Saif founded the first pharma to develop epigenetic drugs and filed the first patents for products that regulate methylation in people with cancer.
Epigenetic kind of therapy space is the next kind of big space in pharma. There's several large companies that all have epigenetic oncology divisions and very small companies as well, startups, that only focus in this area. So it's getting a lot of attention in the recent years. My lab's interested in understanding the role of this epigenetic code in different types of cancers, brain cancers and breast cancers, as well as understanding how this epigenetic code is manipulated during viral infection.
There are epigenetic drugs that are in the clinic now. 2 different types are being used, and particularly in various types of blood cancers, such as leukemias, and many others are in clinical trials currently under development. So that's really exciting, and it's really critical that we continue to develop new drugs. So you might say, well, if we've already got 2 and they work in cancer, don't we have enough? And I will say, absolutely not. There's a huge number of potential targets for these epigenetic drugs, and the more we have, the more we can tailor them to individual patients, to individual diseases.
Because the main criticism is, you're going to do epigenetic drugs, and you're going to reprogram the human, and then his mouth will become a nose, and his ears will become a kidney, and that's not true. That's not true because we know that a lot of epigenetic drugs are very safe. And they're so much safer than the other drugs that are there.
The DNA revolution is totally transforming society. Genetics has become the fastest moving field of science, and in 2017 alone, more than 7 million people had their DNA tested. Once considered outcasts, epigeneticists are suddenly at the center of a whole new world order.
I believe that we will be close to be able to take even placenta DNA as a baby is born and to map those stressors in that DNA that are increasing risk for certain diseases. Is it diabetes, or is it metabolic disease? Is it psychiatry disease? And intervene well before those risks are actually materialized. Now, science has given us amazing tools, both from imaging down to the way we can map now methylation at single base resolution. And we can do it cheap, we can target it. It's amazing what we can do.
You can imagine in another decade, perhaps, you go into the doctor's office and you'll get a readout of your genetic mutations. You will also get a readout of your epigenome and what kinds of changes are present there that we could address through epigenetic drugs.
Epigenetic medicine offers innovative treatment options that are much less aggressive than traditional medicine.
I believe that a lot of epigenetic treatment could be done without using drugs, but by using either behavioral intervention or nutritional interventions, because they all modulate the epigenome. Behavioral therapy could be anything from, a psychological intervention to exercising or to stop smoking, or that's a huge behavioral therapy that worked very well, right? So here is a very good example of a behavioral intervention that had probably more impact than all the drugs in the world. So we can see how behavioral interventions can make a huge difference.
One pioneering behavioral intervention is currently underway with women who were pregnant during Hurricane Harvey.
Potentially 32,000 pregnancies could have been affected by that. So we are piloting an online intervention where women can join the study. Then they go to a website that directs them to do an online writing exercise. It can have very powerful effects for people who've especially gone through trauma. So this is what we're trialing with the Hurricane Harvey study.
Epigeneticists are now using technological tools such as apps to help facilitate the research.
One of the things I'm thinking about is using apps to have what I call a clinical trial in the wild. To do an epigenetic intervention, you see when people use these things, what happens to them. And you get this data fed back, and then you have artificial intelligence analyzing this data. So you'll actually get high quality data from the real environment, not when people are in hospital wards and under control, which is all fake. We have all the tools now actually to do behavior better and to do nutritional interventions better, to measure them with the same rigor that we use to measure, chemical interventions.
Epigenetics offers simple, effective, and safe tools we can use to alter and regulate our genes. Exercise is one of the most effective ways to turn off markers for disease. Even a small amount of regular daily exercise affects epigenetic gene expression in many profound and diverse ways. This is especially important in genes that play a role in insulin response, inflammation, and those linked to a risk of developing obesity or diabetes.
Obviously, exercise has a huge impact on a lot of angles of health, like not just physical health, but brain health as well. It's something that surprised a lot of people, that if you give animals the ability to run a wheel in their cage, their brains are healthier, so they generate new neurons, and those neurons seem to be doing something interesting in the brain. So with exercise, it's a function of not just burning calories that might otherwise be stored as fat that lead to inflammation and things like that, but It can help with stress directly by making stress less toxic, making us better able to deal with stress.
Stress is one of the most critical factors affecting the regulation of our genes.
Stress is a fascinating system because hormone systems in general, like the stress response system, have a way of bringing together a number of physiological systems in a concerted way. So in other words, stress affects the brain, but it also affects the body. It affects the immune system. There are a lot of mental health conditions that are related to stress. In fact, the majority of we call mood disorders, depression, anxiety, are related to how we deal with stress.
Avoiding stress in an increasingly chaotic world seems almost impossible, but our epigenome may offer some form of protection.
Humans are extremely robust. We have withstood a lot of stressors and learned how to come out of it. And probably epigenetics is playing a part in it. The purpose of epigenetic is not to memorize all the bad things that happen and to cause us trouble. The purpose of epigenetics, and probably why it was selected by evolution, is to allow us to deal with the dynamic world, to allow us to deal with stressors. Most people who go into a trauma will come out resilient. So I don't think epigenetic processes are something bad that we need to worry about. They're something good that is protecting us. But we need to identify those who are at high risk and help them. We also need to recognize that most of us are resilient.
Both yoga and meditation are found to relieve stress and promote anti-inflammatory factors in the body. Meditation is renowned for its ability to alter gene expression, and even a few minutes of mindful meditation a day is shown to have beneficial effects on the epigenome. Another simple yet effective way to regulate our genome is through nutrition. Many components of foods have the potential to cause positive epigenetic changes.
So there's a lot of stuff out there, from vitamins to nutritional supplements, that are epigenetic modulators. Actually, you don't need any poisons to modify your genome. Most of the nutritional products that we have around are epigenetic modulators.
There's a lengthy list of foods known to have a positive impact on our epigenome at a molecular level. These foods include leafy greens rich in folate, broccoli and cauliflower, curcuma found in turmeric, and blueberries. It's become increasingly clear that a one-size-fits-all approach to health is an outdated notion.
So I think that at the end it's probably going to be nutrition. It just needs to be focused. I think what we have now, we're probably on the right track, where we say, well, you have a problem, you should go and exercise and eat your vegetables, but maybe we will be a little more focused in terms of what kind of exercise, what kind of vegetables are you talking about? It could be folate, it could be vitamin D, it could be something else, right?
A question that people often ask you is, what's more important, nature or nurture? In other words, the genome or the epigenome. So it really, in a way, is sort of nature via nurture that we're looking at. It's not one or the other, they're linked to each other. In order to be able to maximize our health, we have to look at problems in both the genome and the epigenome, because then and only then will we absolutely be able to really better understand why diseases occur and therefore how to potentially treat these problems.